All right, so today we're gonna do reproduction or cellular reproduction, also called, because there is two types of reproduction. There is the human or the animal reproduction, and then the cellular. Now, as the name says, cellular refers to cell. Cells, what do? Cells, they have to multiply. Why do cells have to multiply? Because we constantly shed or lose cells. We lose cells, so we need to replace them. Now, cellular reproduction. It's asexual reproduction. Asexual, it means that basically offsprings are identical. What does that mean? You start with one cell, and by reproduction, you end up with exactly the same cell. You're not gonna end up with a different type of cell. You end up with exactly the same type of cell. So this is called asexual reproduction. It's called asexual. Sexual reproduction, it basically involves two animals or two living organisms. For sexual reproduction, offsprings have variations. They are similar. You see the difference? Similar versus identical. Asexual, it's identical because you start with the same, you start with one cell and you end up with exactly the same thing. Sexual, it's similar. So if you look at you as a person, you come from, or you were produced by the union of the mom and the dad. Now you look like parts like mom, parts like dad, but you definitely show a lot of variation in the traits or how you look. Now for asexual reproduction, genes come from one parent. Sexual reproduction, as I mentioned earlier, come from two parents. So when you get two parents, you look kind of, but not exactly the same. Now, cellular reproduction can be divided into two types: mitosis and meiosis. Mitosis, by definition, it's division of we call somatic cells. Meiosis is division of the gametes or the sex cells. Now, somatic cells, examples of those ones: cells in the liver, brain, pancreas, heart, muscle. All these ones basically are called somatic cells. And when they divide through this process, which is called mitosis. In a minute, I'll go over both of them. Meiosis, it's basically division of the gametes. Now, meiosis takes place in testes and ovaries. So you're gonna start basically for meiosis with a cell that will end up eventually as the sperm, the final product, the sperm or the egg. Now, what is the difference between meiosis and mitosis? Mitosis, you start with the same number, you start with a number of chromosomes and you end up with exactly the same number. So for example, if I start with 10 chromosomes, I will finish at the end a new cell, or I will get a new cell with 10 chromosomes. If I have meiosis, I start with 10 chromosomes, and at the end, I end up with a cell with 5, or 2 cells with 5 chromosomes. I will end up. So if you notice here, you go from 10 to 5. So basically, if you look, you half the number of chromosomes. In mitosis, you have equal or the same number for the initial and the offspring, for the parent and the offspring. So please just remember what mitosis is and meiosis is. So mitosis, one more time, it's all the cells in the body with the exception of some cells in the testes and ovaries which have the ability to turn or to divide and turn into the sperm and the egg. So if I ask you a cell in your, the cells that make up your hair, sorry, not make up, they produce the hair. What type of cell division they use? Mitosis or meiosis? Of course, it has to be mitosis because it's all mitosis. It's all the cells except in the cells in your testes and ovaries that have the ability to turn into sperm and egg. So please do not forget that part. Now, what happened during this process of cell reproduction? What happens here, basically, the first thing, cells will grow, and then cells will divide. So cells will grow, and then cells will divide. So check this one out here. Let me see if I have room right here. If this is your cell, and you have to make a new cell that looks exactly like that, the first thing that you have to do, it's basically, you are going to take the cell and you have to double it up in size. Once you doubled it up in size, this cell will split up into 2 small cells about the same size. So you start with this size right here. First, it has to double up. Basically, that's what it happens right here. Cells will double up or will grow. Then, after they double up, cells will divide into 2 cells, which could be the same size if it's mitosis, or could be different for meiosis. So this is number 1, growth. Number 2, divide. Now, during the growth process, the most important step, it's DNA duplication. Duplication of DNA, it's the most important step. Why is that? Because DNA, it's the blueprint. So it's the one that's going to tell the cells how they look like. So during the growth process, when these cells will double up in size, the number of chromosomes will also double up. So if I go from 10, let's assume as earlier 10, I start a cell with 10. Once it doubles up, it's going to be 20. Then it's going to be the second step, which is actually the cell division, which will lead to 2 cells with 10 chromosomes. So this is basically how cellular reproduction proceeds. First, it will double up everything, all the contents, including the DNA. And the most important one is doubling up the DNA. Then the actual division, which will have the number of chromosomes back to the initial number. So if you look right here, 10 equal the same one at the end. Now, I keep talking about chromosomes. What the heck are chromosomes? Chromosomes, basically, it's DNA. This would be a chromosome. Now, if you unwind or you break this chromosome into, or untangle it, chromosome looks like this. It starts with DNA that it's really wrapped up, really twisted, and really squeezed into this part right here. That is the chromosomes. Now, how the heck can you squeeze it in there? You get this really long piece, which is the DNA, and you start wrapping up against or around these proteins, which are called histones, which form the chromosomes. So right here, you could see chromatin, it's DNA plus protein. So this one right here, it's the proteins plus the DNA. This is the DNA, forms the chromatin, or it's called chromatin. So this whole thread right here, it's called chromatin. Now this chromatin, eventually it will condense, it will form the structure right here, which is the chromosome. Now what you guys see right here, actually, you see two of the same chromosomes attached by the middle right there. It's called centromere, and together they form this structure which is called sister chromatid. Sister, it means that you have, so you have your, this would be me, and to have a sister would be something that looks exactly the same. It's not called identical sister, but by meaning of sister chromatids, it means that they are identical. So these two, both of them are identical, and they are attached together. So that's why they are called sister chromatids. This is the centromere, and this is the chromatid. Here you could see two chromatids together. When they are united or attached by the middle, by the centromere, they form the sister chromatid. So basically two chromatids attached at centromere, it's gonna form the sister chromatids. The cell cycle, basically, it's the process through which the cell goes during division. Because when it needs to duplicate and then it needs to break down again to form new cells, or divide to form new cells, it goes, or each cell goes to exactly the same process, or the same stages or phases or steps. Now for cell cycle, there is 1, 2, 3, 4, 5 steps. This one will happen every time for or during mitosis. We'll see when we do meiosis, actually this step right here will not happen every time. Now for mitosis, interphase or interphase step, it's the longest one. If you would have right here a whole cycle, this is the cell cycle right here. If you look right here, look how long interphase takes. This is the cell, the initial cell, and look how long of the whole circle interphase takes. So cells spend most of their time in the interphase. The actual division, it's very small. If you look right here, it's very short time, a very, very short time. So during interphase, which is the longest phase, that's when the cells spend most of the time to prepare for actual division, which is very, very fast. So interphase, they prepare. Then they go through these steps right here, which are pretty fast, which are called mitosis or meiosis when we go right there. Interphase, what happens? So here you guys have a bunch of these pictures that I put right here, or photos. So interesting things happen here. Cells prepare to divide, genetic material doubles. So during interphase, genetic material has to double. So there is three phases that take place, or 3 steps on the interphase. So interphase is subdivided into 3 steps: G1, S, and G2. G1, S, and G2. G1, S, and G2. All right, so during interphase, during the G1 part of the interphase, basically the first step of the growth takes place. That's when the cytoplasm starts increasing. So remember, you start with a cell, with the nucleus right in there, DNA in there. During the first step, this part right here, which is the cytoplasm, starts increasing, doubling up basically in size, the cytoplasm itself. While this the cytoplasm start doubling up, there is the S phase, we call it S phase. That's when the DNA actually or the chromosome starts duplicating to this process called DNA replication. So the number of chromosomes in here start duplicating, basically start increasing or turning doubled up or double number. Then it's going to finish up when the cell, it's double the size. I made it too large right here, but it's basically when the cell, it's large enough and it has double the number of chromosomes. So you start with 10 right here. Here, the cell is double in size and there is 20 chromosomes right here. Once the cell is at the end of the interphase, which is comprised of G1, S, and G2, so basically G1, one more time, starts growing, increasing the cytoplasm. S phase, when DNA replication, you go from 10 to 20 or whatever number the chromosomes are. And then there's the G2 growth, when basically it's done growing and now it's preparing for division. Once it went through all these steps and the cell doubled up in everything that's inside, the cell moves into the next phase, which is the actual mitosis or mitotic phase. During mitosis, it's basically division of the nucleus. So mitotic phase is comprised of mitosis, which is division of the nucleus, and cytokinesis, which is division of the cytoplasm, division of the cytoplasm. Here you can see the cells, how they look on under the microscope during interphase. They look like normal cells, and this nucleus right there, it's pretty large. You see the double of number of the number of chromosomes is doubled up. Now, once they get to mitosis, as I mentioned earlier, the DNA, it's double up. The number of chromosome, it's double. Now, that's when the basically sister chromatids are already formed. So remember, we start with 23. If you are a human, you start with 23 chromosomes of different sizes. As you double up these chromosomes, you have double the number right now. You went from 10 to 20 total right now. Now these chromosomes, what they're gonna do? They're going to stick together or they're going to attach. This is called the sister chromatids that I mentioned earlier. Also, during this step, during the formation of the sister chromatids, you can see them, how they look right here. These sister chromatids are attached through this region right here in the middle, which is called the centromere. Now, basically, once the chromosomes look like this, they are ready to move into mitosis. Now, mitosis progresses through a series of stages, which involve prophase, metaphase, anaphase, and telophase. Once this all these stages are done, cytokinesis takes place. Cytokinesis, as the name of it implies, cyto refers to cytoplasm. So that is cytokinesis, that is cytoplasm. So division of the cytoplasm takes place at the end. So let's see what happens on the first step of mitosis, prophase. During prophase, there is this threads that would form right there, this whole thing right here, which will develop from these regions right here, which are the centrioles. This region is called centrosome, and it has these little centrioles things right in there. They are called centrioles. Now from these centrioles, basically this wiring or threads will, or fiber will extend out. Why do they need to extend? Because at the end of prophase, you have to have something like this. And your chromosomes or the chromosomes are attached to these threads right here. These threads, they are called spindle fibers. Another thing that happens in prophase, it's basically chromosomes condense, and then basically they start attaching to and the nuclear envelope also starts breaking down, and then they start attaching to the spindle fiber. Let me see here. Oh, I don't have another picture, but basically during the prophase, chromosome condense, spindle fiber forms, and the nuclear envelope breaks down, and the chromosomes start attaching to the spindle fiber. Next, once they are attached, all these chromosomes gonna migrate to the middle, which is called, this region right here, it's called metaphase plate. That's where the name of the second phase comes from. Metaphase, from the metaphase plate, where basically chromosome lined up at the middle. Then you can see this one's right here. This cell right there, it's actually in metaphase. This phase right here, it's basically in interphase slash starting prophase right here. You see how the DNA is so condensed right here? Here, DNA is still in, I'm sorry, the cell, it's still in interphase. This one here, they also start condensing. So that would be in the interphase slash prophase. So then once they attach to the middle, the metaphase plate, immediately these chromosomes, what they do, they're gonna start being pulled apart. One chromatid on one side, one chromatid towards the other end of the cell. So this spindle fiber start disappearing and start pulling the chromosome, the chromatids, one of the chromatids towards one end and the other chromatid towards the other end. Now, remember, this is very important because if you wouldn't have had that alignment at the middle, it would be very hard to make sure that half of the number of chromosomes will end up in one cell and half will end up on the new cell. Here, obviously, if you look, you can see which cell would be in anaphase. This one right here, this one right here, it's in anaphase. It's very obvious. The last phase, and remember, all these ones happen very, very fast, less than a microsecond, less than a second or something like that. This telophase right here, it's basically start forming the two new cells. Once they reach the end, once they start moving towards the end, this cell membrane here start getting this indentation right in here. It's kind of like a squeeze. Inside here, there is some proteins, they start pulling the middle, the ends of the membrane right there. Now, chromosomes uncoil. Uncoil means that they are not anymore like condensed. And basically the next step, by just keep pulling it together, you're gonna end up with two cells. Nuclear membrane starts reforming, so nucleus forms. Now the last steps, it's basically the cytokinesis when these cells are completely separated into two different cells. So in telophase, this part right here, which is called cleavage furrow, if it's animal cell, cleavage furrow forms, or this indentation forms, nuclear membrane forms during telophase. Now during cytokinesis, the cytoplasm, it's completely broken down and separated into two different cells. Now for plants, this indentation or this pinching on plasma membrane is called cleavage furrow. if you have plants, it's actually, they form a cell plate. And you can see it right here. This is animal cell. It's a pinching. You see how this one pulls together to separate. And for plants, you have these little vesicles that will start dropping this material, which is called cellulose, until what? Until the cell is completely separated. Now, why does it work like this for plants? Because in plants, you have a cell wall. Cell wall outside, it's very, very rigid. Here, you have no cell wall for animal cells. So the actual membrane, it's easier to be pulled towards the middle. Here, it's very hard because the cell wall, it's very, very hard material. So you cannot really pull it. So then you just come and deposit right here at the middle, the material that makes up the cell wall, which is cellulose. And that's when you start breaking the cell. So the process right here, it's called cytokinesis. So if you notice right here, these nucleuses are formed already before cytokinesis starts happening. So the formation of the nucleus takes place during telophase. So the nucleus, it's already formed with the chromosomes. Here you can see the pictures of it right here at the top. Interphase, prophase, they start condensing. This is, look how beautiful this right there, this green one. That is the spindle fiber. You see how these chromosomes start attaching to it. Now, by the end of the prophase, they are just attached randomly. You see how they are attached randomly. Then they align at the middle, which is the metaphase plate during metaphase. Then they start being pulled apart. And then at the end, this process here, since you notice it's being pinched inside, this one actually it's an animal cell. I know it's an animal because it's pinched right here, the cell. It's an indentation. So remember, for animal cells, you have cleavage furrow. For plants, you have cell plate. Cleavage furrow, it's a pinching to indent the membrane so then it can split into two. For plants, there is vesicles, which is those balloon-like structure. They will carry the material, cell wall material, which is the cellulose. You can see right here how in a plant cell, how the actual material is deposited, and then eventually will turn into a complete cell wall. Now, for this process that I just described, this whole cell cycle to take place, first you need some checkpoints. Why do you need checkpoints? Because it's very important for the cell, the initial cell, to look exactly as the last cell. The initial cell, it has to look exactly like the final cell or the last cell or the daughter cell or the offspring, whichever way you want to call it. Now, to make sure that the process takes place properly, there is a need for checkpoints. Now, there is 3 important checkpoints: G1, G2, and M. Now, I already talked about G1 before and G2 something along the way. If you remember, when we talked about interphase, we talked about the cell growing right here. During interphase, we talked about G1, S, and G2 phase right here. Now, actually, G1 checkpoint is in G1 phase or step, and G2 checkpoint, it's in G2 step. M, it's right here during mitosis, the M checkpoint. So let's go over each checkpoint and what do they do, or what do they achieve. The G1 checkpoint. Basically, what it does is just checks the initial cell before it commits to division. So it checks the initial cell to make sure that nothing is wrong with it. So G1 checkpoint checks parent cell for mistakes. Now, the major mistakes that they look for, it's actually to make sure that the DNA is intact or the DNA it's perfect. If they find that the DNA or there is a mutation or some type of change on the DNA is sent to commit suicide. Now, this process through which the cell self-destroys or commits suicide, it's called apoptosis. Apoptosis, it's called. Now, if the cell passes the G1 checkpoint, okay, it's found, great. Now, we found that everything is fine. Now the cell can move into the next step, which is the S and then the actual G2 step. Now, in the G2 step of the cell cycle, we have the G2 checkpoint comes in. Now we want to make sure that after the DNA it's replicated, there is no mistakes. So first you check for mistakes, and then you want to make sure after replication, there is no mistakes. There, check for mistakes on DNA. So first you check to make sure the initial cell has no mistakes on the DNA, and after the chromosomes duplicate, you want to make sure that there is no mistakes on that new replicated DNA, and you want to make sure that DNA is completely replicated. Now, probably you guys hear some genetic diseases in which the DNA or there is parts missing from the DNA. Like if you guys are interested in one of them, check this one out on your own. Cri-du-chat, it's called syndrome. Cri-du-chat syndrome, it's one of the chromosomes, actually it's missing a small piece. Basically what happened, the G2 checkpoint or something happened in G2 checkpoint where it escaped that check. And then it just, division moves ahead. And now the problem is if you have DNA missing from your cells, you might have a syndrome or problem. Cri-du-chat comes from basically kids cry like a cat. That's why it's called Cri-du-chat. M checkpoint, it's a third and the last one. Now, M checkpoint is basically when division pauses to make sure that the chromosomes are distributed accurately to each cell. That takes place basically during anaphase/telophase. That's when this G2 checkpoint, this M checkpoint takes place. So here you want to make sure that the chromosomes are distributed accurately. If they are not distributed equally or accurately, you can end up with another type of genetic diseases. If you guys heard about Down syndrome, people or persons have extra set of chromosome, or they have an extra chromosome 23. So those people actually have 46 chromosomes. Sorry, not 46, 48, because they have an extra set of chromosomes. Now, which one of these three checkpoints right here, which one of these three checkpoints do you guys think it's most important? The most important, and the one that can lead, I mean, all of them can lead to diseases, but in my opinion, the most important one is G1 checkpoint. Why? Because if the cell can bypass, they can bypass this checkpoint right here, that's when the most encountered disease that you hear about takes place. That's when cancers can actually happen. Because cells, what happened? The G1 checkpoint, for some reason, cancerous cells have one quality. They divide very, very fast. You guys hear the cancers grow very fast. Why? Because the cells have the ability to divide very fast, and they can bypass because they can bypass this G1 checkpoint. Now, cancerous cells, as I mentioned, divide rapidly and escape the controls. Now, cancerous cells also spread to all the tissues in the body through blood or circulatory system. Cancerous cells, when the growth takes place, it's called a tumor. Tumors can be benign and malignant. Benign tumors, it's basically localized one. They stay at the original site, like a mole. If you look at on your back, your hands, if you're a white person, you have a lot of moles. Those moles are actually benign tumors. Now, these benign tumors can actually turn into or can become malignant when they start spreading from their initial site to other location. And the process, it's called metastasis, through which malignant tumors spread. It's called metastasis. Now, how do we treat cancers? These days, there is a lot of different types of treatments, but the one that you guys knew in the past, the two main ones was chemotherapy and radiotherapy. Chemotherapy, they basically give you a cocktail of chemicals which will target the cells, the cancer cells, which will replicate. The problem with chemotherapy is that would actually destroy other cells from the body, not only the cancerous cells. And eventually, or it might not even work, or eventually the cancers could return if they were metastatic. Radiotherapy, it's more like localized. They use radiation and they try to destroy the cells at the site where they are localized. Now, the latest type of cancer therapy, it's called immunotherapy. Now, this is the new one. This is a novel method that they use. During immunotherapy, basically, they take your own cells out, if you have cancer, and they reprogram them to actually fight the cancerous cells. So they take cells out and they reprogram them to actually fight the cancerous cells. Now, this new treatment is called CAR-T, C-A-R-T, CAR-T cell treatment, or CAR-T cells, they are called. Cancers, classified basically by their location, and the three main types of cancers: carcinomas, sarcomas, leukemias. Carcinomas, sarcomas, and leukemia. Carcinomas, organs; sarcomas, muscles; leukemias, blood. Now, at the beginning of class, or at the beginning of lecture today, I talked about somatic cells. And I mentioned the cells in the ovaries and the testes. Those are called sex cells. So somatic cells are basically, as I mentioned right at the beginning, are all the cells in the body except the sex cells, Now, what is the difference between sex cells and somatic cells? Somatic cells definitely are non-sex cells, but what is the main difference? Basically, sex cells do not have homologous pair of chromosomes. Somatic cells, all of them, form homologous pair of chromosomes. So this was the chromosome, this one right here that I'm encapsulating, the original chromosomes inside your cells. This is from mother. This is from father, this chromosome right here. Now, during the process of interphase, this mother chromosome gets doubled up or replicated. So now you have two of them. Now, why they're called homologous pair? Because both chromosomes, for example, if this is chromosome 12, let's assume, chromosome 12, remember, we have 23 sets of chromosomes, which is 46 total. So if this would be chromosome 12, you get two sets. You get one from mom and one from that chromosome 12. Now, they are called homologous because they hold the same information, basically, or the same traits. This one right here, both of them could be the eye color. Now the eye color can be different, but it's still the same trait. These genes right here will determine the eye color. this gene above can determine the nose shape. Now, the nose shape, remember, can be different, but it's the same, exactly, the same trait it determines. So these chromosomes are called homologous, and all somatic cells have pair of homologous chromosomes. So as I mentioned earlier, homologous chromosomes have the same length, same information, same centromere position, and the same location of the genes. So if you look right here, you see these initial chromosomes right here, chromosome 12, were exactly the same in size, in location of genes, and everything else. Now, during the process of mitosis or the process of replication, you're going to end up with two sister chromatids. Now, this is for homologous and somatic cells. When you talk about the sex chromosomes are also about the same. And by the way, let me go back here. For somatic cells, remember, we have 46 total chromosomes. So we have 23 pairs. Now, these 23 pairs, you're going to have 23 and 23. This is mom, this is dad. So together you have 46. Each chromosome from here, each chromosome that you got from your mom, it's homologous, or it has the same analog for the dad. Now, when you talk about sex chromosomes, there is actually, for females, there is actually 23 pairs. And at the end, you end up with 46 total chromosomes. Now, this is for females. If you are a male, there is actually 22 pairs, and then there is one sex pair of sex chromosome, one sex pair. Now, males are different than females in one thing. Their sex chromosomes are different. Why? Because we have an X and a Y. That's how we label. For females, you have 23 pairs. Same thing. 22 plus 1 sex chromosome. But for female, the sex chromosomes are X and X. They are exactly the same. So that's why I call them 23 pairs. For male, there is 22 pairs. And then there is the sex chromosomes. Now, these 22 pairs are called autosomes. Same thing right here. For females, all of them could be considered autosomes. There's still also 22 autosomes only. Autosomes. There's still same 22 pairs. They are autosome. And then the last one is the sex chromosome. Even the sex chromosomes for female are the same. For male, they are not the same. Now let's go over meiosis. So, quick review. Mitosis, basically, so let me write it right here. Mitosis, so far we learned, you start with one cell. Let's assume 23 chromosomes. This cell will double up in its size. So now you're gonna have how many chromosomes? 46. And then eventually you end up with two cells, the same size as the original one, 23 chromosomes. Meiosis, it's a little bit different because at the end, you end up with half the number of chromosomes. So right now, I start with 23. Oh, shoot, it's not gonna work really well because I need to halve the number. So let me make another one. I start with, let me put right here, 46. you go to 92, and then you end up with 46. So we can make a human being right here. So you start with 46. So right here also, I start with 46 during meiosis. During the next step, they're gonna grow, double up in size. So I end up as 92 chromosomes. So if you notice, it's the same thing as mitosis. It's almost the same thing as mitosis. Now what happens here, the cell will divide, and you're gonna end up with two cells the same size as the original one, 46 cells. Now this one here, during meiosis, there is no more duplication. What happens right here, this one, or these two right here, would go directly into the next step. There is no more duplication step. And they will divide into two smaller cells, which are the sex cells, which have 23 chromosomes. And this one here, it's the same way, 23 chromosomes. So if you notice right here, mitosis goes through one division. Meiosis goes through two divisions. Now, there is one duplication step. Duplication. And here on the second step, there is no duplication. Now, for mitosis, there is no such thing as the next step. Now, this one right here, through which you get the first step, it's called meiosis 1. So all the way here, it's called meiosis 1. From here to here, it's the second step, and it's called meiosis 2. Now, all cells, when they start with a diploid number, or a set, or two sets, whichever one you want to call them. 46 chromosome, remember, was 23 mom, 23 dad. Now this diploid, it's called diploid because it has two sets, 2n. Haploid number, it's one set of chromosome. So when you look right here, the beginning, it's 2n. Here at the end, it's also 2n. 46, 46. Here you have 2n. At the end of meiosis 1, you also have a diploid number. And at the end of division, you actually have a n. So you go from diploid to haploid. Now these cells right here, if you notice in mitosis, you start one cell, you end up with two of the same cells. In meiosis, we start with one cell and we end up with 4 cells right here, which are completely different, and they have half the number of chromosomes. Now these cells could be identified as the sperm and the egg. Now meiosis occurs in the sex organs. Which ones are those two sex organs that produce the gametes? Mentioned earlier, testes and ovaries. Here you can see the process. Here you see two naked people. Now I notice that they dress them up in the book to be politically correct. Then they go through meiosis in the ovaries and the testes. And then what's going to happen? End up with the sperm and the egg. The sperm and the egg will fertilize, and you end up with a zygote. Zygote is the first structure that all of us were at one point. This structure basically has the ability to divide. This one cell here has the ability to divide into millions and gazillions of cells and turn into these two individuals again. The process through which the cell divides in the testes to form sperm, it's called spermatogenesis. The process through which the female cell divides, it's called oogenesis. Now when the sperm and the egg come together, they reform the zygote, which has a 46 total number of chromosomes. So remember, the egg and the sperm, both of them have 23 chromosomes. Once they come together, you end up with a zygote, which will have a 46 set of chromosomes. Now, as I mentioned earlier, meiosis involves 2 steps, meiosis 1, meiosis 2. It's very, very similar to actual mitosis. There's just a couple differences, which I'm going to point them out while I go over. First, the steps are exactly the same. You have interphase, which is right at the beginning. It's the one that the cell spends their time, most of its time. Then you have the same 4 stages or steps: prophase, metaphase, anaphase, telophase, and cytokinesis at the end. Now, since I showed you here, there is two main categories. There is meiosis 1, the main stages or categories, meiosis 1 and meiosis 2. Remember, in meiosis 1, you go from duplicate them to 92 and then go back to 46. This is the first meiosis. This is meiosis 1. Now, this one will move on to the next step, in which it goes to 23. How many? There is 2 of them. Total is going to be 4. So this is meiosis 2. So remember, here you start with 1 cell, you end up with 2 cells at the end during mitosis. Meiosis, you start with 1 cell and you end up with 4 of those gametes. Now, the main difference between meiosis and mitosis, the steps or the things that take place, one of the major one, it's prophase 1. Please remember this one. There is 2 things that happen majorly here, different than actual mitosis. First, homologous chromosomes pair up and they form this structure which is called the tetrad. Also, chromatids exchange parts. This is called crossing over. Now, when I showed you at the beginning, and you can go back and look at it, for mitosis, when those spindle fibers form, chromosomes attached like this. This is in mitosis. They attached individual, the sister chromatids. When we talk about meiosis, you have the same spindle fibers, chromosomes attached together as homologous. This structure right here, it's called tetrad. Tetrad means 4. So there is 4 actual chromosomes or chromatids. Now, crossing over, what happens? Basically, this sister chromatid can exchange arms. These sister chromatids can exchange arms, which means what? This piece right here can replace this piece right there. Let me see if I have a color, different color right here. So this piece right here, you start with this chromosome right here. Let me put it this way. So you start with this chromosome. Let me erase it first. Should I can erase this damn thing? Okay, let me go back to red. Okay, I cannot erase it. Let me see, I can erase them, but let me do them then differently. So this chromosome, let's just assume this was the chromosome, right? Oh, shoot. Okay, let me do this one. So this was the initial chromosome. This was the father. And this one here is the mother chromosome. Now what happens here, these chromosomes overlap. So now the mother chromosome can pick up this is the father chromosome. So the father can actually acquire this little piece right here. they can exchange their arms. So basically this red here, this black here becomes red. This red becomes black. And that's what actually it happens during crossing over. So now let me see. Oops, this thing doesn't work anymore. Let me see, clear all drawings, okay. Oops, let me see, is it working now? So now, okay, I don't know what happened here, but let me go to the, oops, what happened here? Can't share anymore. Oops. So let's see what happened here, sorry. During prophase 1, they form the tetrads. Also, they cross over, you see right here? You see these parts right here? Check out here. Oops. Once they crossed over, you see this blue is replaced with green, and this green is replaced with blue right there. This process here through which they replace each other, it's called crossing over. Now, this is the process that leads to variation. Because as you're gonna go through these steps right here, let's go quickly over them. Formation of the tetrads, crossing over. Now, they will attach to the spindle fibers as you see right here. Then they will get to the metaphase plate. They will align at the metaphase plate. Remember, this is still meiosis 1 that we're talking about. So prophase 1, chromosomes will condense, chromosomes will stick together, form the tetrad, nuclear membrane will disintegrate, then the sister chromatids form, nuclear membrane will disintegrate, then the chromosome form the tetrads, they cross over, and they are also attached to the spindle fiber in prophase 1. Metaphase, they just align as homologous chromosomes. And then what they're gonna do, they're gonna get pulled apart during anaphase, exactly the same thing as mitosis. And then they form the cleavage furrow if it's an animal cell. Now you end up with 2 cells. You end up with 2 cells right there. As you move into meiosis 2, they go or undergo a second division. Now, there is no more replication taking place. So there is no interphase. So interphase 2, it's gone. There is no more duplication. Why? Because as they go through these steps right here, and if you notice, prophase 2, it's exactly as mitosis. Spindle fibers reform, chromosomes reattach to the spindle fibers individually, as in mitosis. Now, when you look at these chromosomes, they look different between each other. Then they align at the metaphase plate, then they split apart, and then eventually they go through telophase and cytokinesis. So you end up with 4 cells. Now, when you look right here, look at how these cells look. And let's go back to, prophase 1. We started with these ones here. You see how the green with green, blue with blue, green, green, blue, blue. There were 4 chromosomes when they started. 1, 2, 3, 4. These are the 4 sets because they're already duplicated. But there is 4 chromosomes. 2 blue, 2 green. 2 blue, 2 green. Now check out right here what we have at the end. You have only 2 chromosomes. So you start with 4 and you have 2 chromosomes. Now, when you look at them, they look different because of the crossing over. Now they have different pieces right there attached to them. Now, that's why when you look at your children, or when you're going to have children, they look so different than you, because the sperm and the egg has this process of crossing over. And I show you at the end my 23andMe if you guys are interested in seeing it. So quickly, this is the whole overview of meiosis. Remember, the most important step, the critical step, it's the prophase 1, formation of tetrads and crossing over. Then everything else, it's the same. Also, critical to remember, there is no interphase in meiosis 2. There is no interphase. Remember, there is no duplication. So all the way here chromosomes are duplicated. Here, if you notice, they are just individual. There is no more duplication taking place right here. You have the same chromosome passing into the next phase. Here, you started with 1, 2, 3, 4, 6. So basically here there is 6 chromosomes at the beginning. Then you end up with 12. 2, 4, 6, 8, 10, 12. There's 3 sets of chromosomes there. You see when they align at the metaphase plate, or you see when they form the tetrads, they are homologous chromosomes sticking together, the same 2 ones. You see how they stick together? Now when you look right here in the metaphase 2, they are just individual. Also, you see here these reds to the blues and the blues to the reds. That's basically crossing over because of the process of crossing over. Now, what are the main differences and similarities between mitosis and meiosis? Similar, basically, there is only one duplication of chromosome during that single step, which is interphase. There is no interphase 1, just interphase. There is only one in both of them. Which ones are unique to meiosis? There is 2 divisions, meiosis 1 and meiosis 2. Pairing of homologous chromosome, which is formation of tetrads. Exchanging of genetic material, which is called the crossing over. What is the outcome of each process? This is basically kind of like a mini review. Mitosis, you end up with 2 genetically identical cells with the same number of chromosomes. Meiosis, you end up with 4, because remember, there is 2 divisions, and the second one, there is no duplication. And you guys can see right here, this is the parent cell, 1, 2, 3, 4, not a picture of it. As it goes through mitosis, it doubles up. And at the end, you end up, these 2 are the same. Meiosis, first double up, then they form the tetrads. Here, there's the crossing over, tetrads and crossing over. Then they align, if you notice right here, they align as sister chromatids as homologous chromosomes. Then they go through meiosis 1, and at the end, check out, do they look the same? They don't look the same. Look, this one is all blue, this one here is blue with red. Now, as they continue dividing on the second step, this is the final product. Now check out these ones, these 2 right here. Here you started with 4 chromosomes. Here you end up with 2 chromosomes in each cell, 2 chromosomes in each of the cells. So please review it, and if you have any questions, you ask me. Now, as I mentioned earlier, if mistakes happen during those checkpoints, you end up with certain diseases. We call them abnormal number of sex chromosomes. Now, 3 diseases or 3 genetic diseases that I want you guys to remember forever and for life. Down syndrome, which probably you guys see there, or you see children. They have a special feature like face feature, and basically they have a little bit more of mental retardation. So why is that happening? Because for these people with Down syndrome, there is trisomy or an extra chromosome 21. So this is the karyotype. When you look at it, this is called karyotype. And it shows you how many chromosomes, total chromosomes we have. We have chromosome 1, you see this is mom, dad. That's how our chromosomes kind of look like. They are exactly the same size. These 2 chromosomes are called what together? They're called homologous. So we have 23 pairs. If you look right here, this is a female. Looking at this one, sorry. Oh no, this is a male. I'm sorry. This is a male. Why? Because there is an X and Y chromosome right here. So this male here has 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. 22 sets of homologous chromosomes, or 22 pairs of homologous chromosomes. This pair, it's not homologous because this is a male. Now, if you look at the Down syndrome, it has an extra 21 chromosome. Turner syndrome is another one. Now, this one is very interesting. You're not going to see it. There is no problem with the, in Turner syndrome, it's in females. It means that it's an X and O chromosome. Now, when they refer to an X and O, they refer to sex chromosome. Now, the only problem or the only difference between a normal female and a female with Turner syndrome, it's basically the ovaries or the female is sterile, cannot have babies. That's all what it happened. Now, there is other features that were observed, characteristics for this Turner syndrome female, but they're not very visible. And it's very hard to identify a female with Turner syndrome just walking around campus or walking on the street. They have a little web neck, which is kind of like this. They have, the breasts are not developed, they're less developed. And sometimes they have long arms. For males, there is the analogy of Turner syndrome in female, X and O. Now for male, can be X and O, it's X, X, and Y. So basically it has three chromosomes for the sex chromosomes. It has 3 of them. Now, same thing. If you look at a male with Klinefelter syndrome, you're not going to identify, you can't tell me. They have a few characteristics that might be observable, but not necessarily means that they have it. But there is one that it's the main problem, which is also the testes, or the testes cannot produce viable sperm. So they are sterile, they cannot make babies. Now, other features as in the female case, they have no facial hair, but it doesn't mean that necessarily if you don't have facial hair that you have Klinefelter syndrome. So please remember that. They have some breasts. Somehow the breasts start developing, probably because of the second X chromosome there. They have very long arms and very long legs. Very long arms and very long legs. That tells you that you would have, or that person would have, Klinefelter syndrome. Now, don't go around and just look for people with long arms, long legs, and start labeling them. Oh, you have Klinefelter, or, you know, I had a student a few years back that walks around campus and comes back and tells me, oh, professor, there's a lot of students, a lot of girls here with Turner syndrome. And I'm look at him and I'm like, what do you mean by that? Oh, you know, they have small breasts. That's what you told me. And the neck was kind of wet. And I looked at him and I was like, you can't go by those features. You cannot identify them by those features, okay? So this is all about the lecture today. And here at the end, I'm just gonna show you, I saw this is a Pulitzer Prize winner. And here it shows you a HeLa cell line dividing. Now this HeLa cell line basically are very specific, very interesting cells. They were recovered from a person that was clinically dead, but for some reason her cells were still dividing, even she was declared clinically dead. So here you can see the actual meiosis. That's what it happens right there. So this was a picture that was taken by, I guess, a photographer under microscope, and I thought it looks really, really cool. This is the cells when they'll divide. And I guess this is all.